Welcome everybody to our VacCAP webinar series. My name is Amaya Tucha and I'm part of the VacCAP team, and I'm going to be the moderator for today's webinar. Today we will be discussing the genetic control of anthocyanin content in blueberry with Dr. Sara Montanari, who is part of our international collaborator team from New Zealand. Dr. Montanari is a molecular breeder at Plant and Food Research in New Zealand. She works on the implementation of genomics into breeding, mainly in horticultural crops such as blueberry, raspberry, pears, and hops, but also in aquaculture species. She received a PhD in plant molecular biology in 2015 from the University of Angers in France. Her PhD work focused on the genetic mapping of pest and disease resistance in pear, and it was a collaborative project between Fondazione Edmund Mach in Italy, the INRA of Angers, and Plant and Food Research in New Zealand. For her postdoc, she moved to the University of California, Davis, where she continued working on pear breeding and genomics. Dr. Montanari has been a scientist at Plant and Food Research in New Zealand for the last 4 years. We are very excited to have Dr. Montanari as a speaker today, and I just want to remind everybody to keep yourself muted and please your cameras off so we don't distract our speaker during her presentation. If you have any questions, just go ahead and just type them either on the chat box or the Q&A, and we will be addressing as many questions as possible at the end of the presentation. And with that, I'm going to turn it over to Dr. Montanari to start her presentation. Thank you.
Thank you, Amaya, for the introduction. I'm going to start sharing my screen now. All right, can you see the presentation all right?
Yes, perfect.
Okay, thank you. Well, good morning, everyone, or good afternoon, or good evening, wherever your time zone is. So today I'm going to talk about a work that we've been doing at Plant and Food to understand the genetic control of anthocyanin content in blueberry. And I'm also going to present some of the results from Massimo's group at North Carolina State University, who did the work on the same traits in parallel with us. So start with a bit of an introduction about the study. Obviously, you don't need an introduction about blueberry with this audience, so I'll just go straight in with the anthocyanin. Anthocyanin are compounds that have received a lot of attention lately because they have been linked to a beneficial effect for human health. And these include, let me put the laser in. These include antioxidant actions, anti-inflammatory, anti-proliferative, anti-obesity, and neuroprotective actions. And they're also responsible for the coloring, and in blueberry in particular, they're linked to the red and blue pigmentation. So now I'm not a biochemist, but for this project, I had to learn a little bit about the molecule of the anthocyanin in order to be able to do this study. And now I'm going to try to explain it to you here, because it's quite important to understand the rest of the webinar. So here there's an example of an anthocyanin molecule. It is composed by two main parts. This part here is called anthocyanidin or aglycone moiety. And in blueberry, there are 5 different types of anthocyanidin, and these are the cyanidin, delphinidin, peonidin, petunidin, and malvidin. And then there's a sugar moiety linked to the aglycone moiety. And in blueberry, we have mainly 3 types of sugars: glucose, galactose, and arabinose. Sometimes we also found the addition of an acetyl group to this sugar, in particular to the glucose and the galactose. So this brings the number of possible sugar moiety to 5. So in blueberry, we can find 25 different types of anthocyanin that are the composition of these 2 different moieties. So the objective of our study was to first build a high-density, high-quality genetic map for highbush blueberry. This was not a trivial task because blueberry is autotetraploid. And then we did a characterization and quantification of the anthocyanin in dry blueberries and used this data to identify QTLs that control the anthocyanin content. So this is the mapping population that we used at Plant and Food Research. It's a cross between these two cultivars, Hort Blue Petit, which I also call M7 for brevity, and Nui. And we have about half of the population coming from M7 as a father and Nui as a mother, and the other half of the population is the reciprocal cross. For the genotyping, we did whole genome sequencing on the two parents using 10x Chromium. And then we did capture-seq on both the parents and the offspring using the older rapid genomics baits. So we look at about 280 offspring, and after proper filtering and dosage calling, we ended up with a SNP set of about 50,000 SNPs. So as I mentioned before, mapping in autopolyploid is not a trivial task, but it's been a little easier lately because there's been like a lot of tools that have been developed and published to work on autopolyploid species like blueberries. And for this work, we used these two R packages that were developed by Peter Burke, Polymap R for the linkage map and PolyQTL R for QTL mapping. So in Polymap R, we were able to build consensus linkage map for the two parents, which you see here. So you can see we had 12 linkage groups that correspond to the 12 chromosomes of blueberry, and the map is very high density. We had about 80,000 SNPs over 1,700 centimorgan. With that software, we were also able to do the phasing of the map. And here I'm reporting an example for linkage group 1. You see in the middle, we have the consensus linkage group for the two parents. And on the side, we had the map for the 4 homologs of each parent. So here on the left is M7, and here on the right is Nui. And this is very important to be able to perform QTL mapping afterwards. We did also some quality checking of the map, beginning with the quality checking feature of Polymap R. So the software returned plots that look like this that basically help us identify location in the map where there was possible mismapping or some noise that we could potentially fix. And after that, we did a final quality checking looking at Murray plot. So here again, I'm reporting an example of a Murray plot for linkage group 1. Each dot represents a SNP. And this is a plot of the physical position of the SNP on the x-axis versus the genetic position on the y-axis. And so what we expect is to see sort of a linear correlation at the beginning here, then a plateau that usually corresponds to the centromere, and another linear correlation for the second arm of the chromosome. And so we did this work for all the 12 linkage group, and everything looked good. So we were quite happy with the quality of the map. Now for the phenotyping, we started by harvesting blueberries from all the 300-something offspring that we had and the two parents. And we harvested blueberry at the stage 8 or eating ripe stage. We froze them and juiced them and did HPLC mass spectrometry. And in the end, we were able to extract information about the anthocyanin content and composition for each of the offspring and the parent. So as I mentioned before, in blueberry, we have 25 possible types of anthocyanin that correspond to the combination of 5 types of anthocyanidin and 5 types of sugar moieties. And we found all of them in the mapping population. So for the follow-up study, I used the concentration of all these 25 individual compounds. But I also look at total concentration of anthocyanin grouped by either their common anthocyanidin moiety or by their common sugar moiety. And I also use the total anthocyanin concentration. We had 2 years of data, 2019 and 2020, and the data were from medium to highly correlated, depending on which specific anthocyanin we look at. In the second year, in 2020, we also collected some additional data at harvest because we wanted to see if there was something else that had an effect on the anthocyanin concentration. So we look at what I call here bush maturity, which is basically a measure of the average ripening stage of all the berries on the bush at the time of harvest. Then we measure fruit diameter, fruit weight, and we recorded harvest date. And here I'm showing a correlation plot between among all these four harvest data that we collected and the concentration of all the different type of anthocyanin. And I think the information that we get that was more important for us is what I'm highlighting here with these two arrows, which is showing basically that the fruit diameter and the fruit weight, which are proxy for fruit size, are significantly and negatively correlated to the concentration of all different type of anthocyanin. So this was expected because we know that anthocyanin in blueberry are found mainly in the skin. So for same weight of blueberries, you have more skin and some more anthocyanin. But it also was important for us to measure this data because we wanted now to correct the phenotype for fruit size. And so that's what we did with the 2020 data. We then run a principal component analysis using the data from the concentration of the 25 different type of anthocyanin. So here I'm showing an example of a PC1 versus PC2 plot for the 2020 data. Each dot here represents one of our offspring. And so you can probably see that the offspring separate into two main clusters along the PC1. And so when we went to look into which were the specific anthocyanin that were mainly contributing to the PC1, we realized that all the anthocyanin that contain, excuse me, that contain acylated sugar moieties were the one that were mostly contributed to the PC1. We also investigated the PC2 and realized that anthocyanin that contain the sugar, galactose, and the ribinose were the one that had the major contribution to the PC2. Overall, what this is telling us is that the sugar moiety is the part of the anthocyanin molecule that is more important for the variability in this case, and in particular, the presence absent of the acetyl group in the molecule. In this slide here, I'm showing comparison that we did for the two parents of our mapping population. We wanted to look if there was some difference in terms of specific anthocyanin composition for the two parents. So this bar plot here show, each of them show the parent whole blue petite or M7 on the left and the parent Nui on the right. On the left-hand side, we have anthocyanin composition with anthocyanin grouped by their sugar moiety, while on the right-hand side, we have anthocyanin grouped by their anthocyanidin moiety. And then we have 2019 data at the top and 2020 data at the bottom. So the first thing that we notice is that the total amount of anthocyanin in the two parents, so basically the top of the bar, is not consistent between the 2 years. So in Haute Blue Petite, we have less total anthocyanin in 2019 than in Nui, but the opposite was observed in 2020. However, we can notice that the patterns in terms of composition of type of anthocyanin between the 2 years were more consistent, both for the two type of groups that we look into. When we look in the anthocyanidin grouping, we notice that there's not really much difference between the two parents, so that doesn't seem to be very interesting. But when we look at the anthocyanin group by the sugar moiety, we notice that there are different patterns between the two parents. First of all, for example, we see that Nui has about a third of anthocyanin that carry the glucoside moiety, and those are mostly absent in Haute Blue Petite. And on the other hand, in Haute Blue Petite, about a third of the anthocyanin carry the acetyl group, in particular acetyl linked to galactose. And these, again, are mostly absent in NUI. So again, as for the PCA, the conclusion here is that the sugar moiety is the most interesting part of the molecule, and the presence absent of the acetyl group is also very interesting. Now, for QTL mapping, I use the concentration of all the single compounds, so the 25 type of anthocyanin, and then I use all the total values, so that's include total anthocyanin, but also the sum for all the anthocyanin group by either their sugar moiety or their anthocyanidin moiety. And then I also use the PC1 and PC2 values. And I had data for 2019, 2020 adjusted for their fruit size, as I mentioned before, and we also used the estimated marginal means for the 2 years. Excuse me. So in total, we found, for all this data, we found 166 QTLs across 6 or 7 linkage groups, but there are 2 linkage groups that show major hotspot of QTLs, and these are linkage group 2 and linkage group 4. So for this hotspot, the QTL were consistent across different compounds and stable across years. Also, many of the traits were associated with both QTLs. When we look at the QTL that we found using the PC1 and PC2, we noticed something interesting. So the PC1, as I mentioned before, was mainly linked to the presence-absence of the acetyl group in the molecule. And using the PC1, we found only the linkage group 2 QTL, and it was very, very strong. When we used the PC2, which was linked to the presence of galactose or arabinose in the anthocyanin molecule, we only found the QTL on linkage group 4. When we look into the region, the confidence interval of the QTL, we were also able to identify some candidate genes. We found 4 interested genes in the linkage group 2 QTL, and there were 2 acetyltransferase that seems to be particularly interesting, since this QTL seems to be linked to the acylation of the molecule. And then we found 31 candidate genes in the linkage group 2, and some of them were also linked to the glycosylation. Now, in parallel with our work, Massimo's team did something similar at NCSU, and they started looking also at the anthocyanin genetics in blueberry using their population Draper by Jewell. They had 3 years of phenotypic data and found 188 QTLs. And also, they also found, like us, major hotspots, particularly on 4 linkage group 1, 2, 4, and 8. Similar to our results, the QTLs on linkage group 2 were linked to the acylation, while the other 3 were linked to the glycosylation. So when they look into this major QTL region, they also identify some candidate genes, and they even run some RNA-seq data to kind of have a first step of validation for these candidate genes. So in linkage group 2, which was linked to the acylation, they found acetyltransferase genes that seems to be the best candidate. In linkage group 4, they found genes that were linked to the ratio between glucoside and presence of galactoside or arabinoside in the molecule, and they found glycosyltransferase gene in this group, which was very interesting. And finally, they looked at linkage group 8 and also found genes that were linked to the glycosylation. And interestingly, this QTL also overlap with a QTL for soluble solid content. And so there were genes that were responsible for sugar transport that are seems to be particularly interesting for this region. So thanks to VacCAP, our group and Massimo group were able to communicate and share results, and we compare our QTLs and realized that the QTLs on linkage group 2 and 4 were overlapping. So here I'm showing chromosome 2 on the left and chromosome 4 on the right. And the QTLs that we found at Plant and Food are in blue, and the QTLs found by NCSU are in red. And so you can see that for both chromosome, the QTLs overlap. So this is the first step of validation of our results. And both groups also identified candidate genes in these two regions that are particularly interesting, including acyltransferase gene in chromosome 2, a glucosyltransferase gene in chromosome 4. So now at Plantain Food, we decided to focus on the QTL on chromosome 2, because that was the strongest one for our mapping population. So this is the QTL that is linked to the acylation. And here I'm showing a plot that was returned by the polyQTLR package. So basically that package allowed us to identify which was the specific haplotype that had a positive effect on the trait. And we realized that consistently haplotype 3 of Hort bleu petit, or M7, was linked to an increase in concentration of all anthocyanin that contain acylated sugar. And the box plot here shows the same thing. So the first thing that we wanted to do was actually to dive deeper into this region and try to identify what was the causative variance that is linked to this trait. To do that, we did some PacBio hi-fi sequencing of one of the offspring of the mapping population, and then we used a trio binding approach to do haplotype assemblies. So how this worked, basically we had Illumina reads for the two parents that were used to generate K-mers that were separated into two sets, one for M7 and one for Nui. And then these K-mers were aligned to the progeny hi-fi reads so that the progeny reads could also be separated into two groups derived from each parent. We then did a separate genome assembly for these two groups of reads, and we were also able to resolve them in haplotypes, thanks to the variation among haplotypes within the parents and the recombination from parents to offspring. So in total, we had 4 haplotype assemblies for the progeny, 2 from Nui and 2 derived from parent M7. And you can see that the progeny carries the haplotype 3 from N7, which is the one that we are interested in, that carries the that was linked to a positive effect of the trait. We also did some gene annotation of this region and using the sequence of the two that derive from the M7 parents, and we found 6 copies of acetyltransferase gene in the haplotype 3 and 5 copies in the haplotype 2. And we started some work to do some functional validation of these genes, and this include a transient assay in Nicotiana bentamiana, where we tested one of the copy of the acetyltransferase using also MIB-A to create anthocyanin. And then, so when we extracted the tissues and ran them through mass spectrometry, we could identify that there was generation of anthocyanin that contain the acetyl group. Excuse me. So in conclusion, our work in collaboration with Massimo's group released 2 high density and high quality genetic maps for blueberry, which is autotetraploid. We also had a detailed anthocyanin profile for highbush blueberry population, which allowed us to identify novel QTLs. We had 2 major QTLs on linkage group 2 and 4, and these were stable across years, and they were not affected by fruit size. And these results were validated across different genetic background using the population at Plantain Food and CSU. So at Plantain Food, we have been focusing on the QTL on linkage group 2, which is linked to the acylation. And this is very interesting because the addition of an acetyl group to the molecule increases its stability to heat and light exposure. And so it's more desirable for the pigment and food industries. We identify candidate genes in the QTL confidence interval of linkage group 2, 4, and 8. And we have work in progress to functionally validate the acetyltransferase gene in chromosome 2. And we're also doing some work using phase genome assembly of a progeny of our mapping population at Plant and Food to identify which is the causative variant of this QTL. And so eventually we can use this information to design marker for marker-assisted selection. So this work resulted into 3 publications. First 2 publications were released last year into a special issue in Frontiers, one from the work from Plant and Food Research and work from NCSU, and they were published as sister papers. And then together we work on a review that was just recently published in Plant Physiology. Obviously, there's like a lot of people that were involved in this study, and I'm acknowledging them there, and I'm acknowledging Massimo and Molla for the work, and here the funding that allowed us to do this study, and all data have been deposited on the GDB. And thank you for your attention. Happy to take any question now.
Great, thank you, Sara. That was a great presentation. I don't see any questions right now in the chat, but if you have any questions, I think we have enough time that you can either write them on the chat, or if you want to unmute yourself and maybe ask the question, you're welcome to do that too. So we'll just wait a couple of minutes to see if there's any specific questions. I'm gonna paste here on the chat also, we're recording this webinar, and so this webinar will be available on our YouTube channel, and here's the information on the chat.
Can I ask a question?
Yes, please, David, go ahead.
Are you nervous, Sarah? It's actually a question not necessarily for Sarah, but for the VacCAP community, because we're zooming very, very hard onto the causative variant for that chromosome 2 QTL. And I was wondering, what sort of resource do we have to, validate those causative variants? So this functional analysis that's underway, but what about, the validation of the perfect marker for that particular trait? Do we have panels of diverse accessions or breeding accessions that have been typed and phenotyped to be able to validate the putative variants, the causative variants that we identified? But it's more like a question to Massimo and the rest of the project team rather than Sara. So you're off the hook, Sara, anyway.
I can say, yeah, we do have a diversity panel that has been validated, has been evaluated, and we can have some conversation. It's quite diverse, that could be used to validate some of the variant that you guys detected. And it's on top of the QTL that we of the gene within the gene.
Great.
I don't know if there is
Massimo.
anybody else that has done any work on anthocyanin already, that it's on the call.
If there's anybody,
I have a question,
Dr.
Attuccia. I have a question for Dr. Montanari.
Go ahead, Fernando.
Yes, a great presentation, by the way. I had at the end, you talked about how you were going. The next step then is marker-assisted selection, and so my question is, what kind of performance gains do you expect upon implementing or like stacking or fixing your alleles for the QTL? Yeah, that's pretty much my question.
Yeah, I don't have an answer yet, because this is just the very first results that we're having. So this is a bit linked to what David was asking. So our first thing is to identify this causative variant and design the marker. But then we need to validate this marker to begin with before being able to use it for marker-assisted selection. So we need to look into different genetic background to see if this marker actually is linked to the trait that we're looking at. And then I'm not sure about stacking. It looks like this particular QTL, it seems to be linked to the specific acylation of the molecule. And so it's a very specific point in the biochemistry pathway. And I'm not sure if there are any other region that might be linked to this trait or that would make us able to do some stacking with other allele. Also, the QTLs for us in this region was very, very strong. So it seems to be monogenetic. So I don't know. That's my answer right now. It's just something that we need to explore a bit more.
Sara, I would just complement your answer for Fernando. So we have also done a study where we looked at the effect of the structure of the anthocyanin on the bioaccessibility of the anthocyanin. So in general, anthocyanin are very, very low. When we eat anthocyanin, a very small fraction are absorbed in the gastrointestinal system. So, but what we found is that using in vitro system is that the acylation increase the bioaccessibility of the anthocyanin. Technically, could increase the absorption of the anthocyanin. So that could be a possible a trait to look at, increasing the bioaccessibility and bioavailability of the anthocyanin for health reason.
Yeah, thank you, Dr. Urizo. I appreciate that.
All right, thank you. We have a question in the chat. Ibrahim is asking, how did you use the principal component in the QTL?
Just when you run a PCI, you have you return the value of PC1, PC2, and all the other PCs. So I use those values as a phenotypic trait, basically. The interest in doing that is that when I was looking at the total, I knew I was not going to be able to, they were not being representative of the proportion of the different anthocyanin, while the PC was able to represent that. And so that was why I was interested in using this data.
Great, thank you.
Sara,
anybody else?
Thank you for a nice seminar. A question. So do you know, do you have any sugar data or any even salt content, BRICS data on this population to see the correlation? Because the QTL, you found a strong QTL, the glucoside group, the addition, to see if that affect the use of the sugar as a substrate. And basically removing the sugar from being available in the, of being soluble in the fruit. You don't have any of those data?
We don't have any. No, we have been discussing, like, yeah, it's something that is on our radar to look at other, sugar and also other compounds for this population. Yeah, that would definitely be interesting.
But we did find some correlation with the soluble salt content. That's why.
Yeah, we did find a minor QTL on linkage group 8 as well, so that would be quite interesting to look at.
Do we have any other questions?
Since nobody, or there are none, I have another one on the genome assembly.
Oh, everybody's being really shy.
Yeah, on the genome assembly, basically these, so if I understood correctly, each of the two haplotype are, so you assembled 4 haplotypes and then you use the K-mer to phase. So basically you reconstructed the 2 haplotype of one parent and 2 haplotype of the other parent. Is that correct? Coming from one parent from another parent?
Yeah.
Okay.
I can chime in there. It's actually, my name's Ignacio. It's the other way around. You use the K-mers to bin the hi-fi reads, and then you do 4 different assemblies just from the binned reads.
Okay.
So each haplotype's assembled independently.
Okay, that's great. Thank you, Jose.
Yep.
I have one more question, Dr. Montanari. So I was looking at your article, and when you were doing the high performance liquid chromatography, what kind of variation did you see in the fruit, or do you feel like it was pretty consistent between individuals in the population? I'm just asking for my own, because it says here you needed 20 grams per sample. And so suppose you have an experimental population where you don't have 20 grams. I mean, do you foresee, yeah, I guess it's more of a general question for me in my own experimental populations, just to how I guess my question is, yeah, does this approach account for variation in the fruit that you see across your populations?
I'm not sure I'm able to reply to that. I have to say, like, because I didn't do the analysis. What I want to stress out is that we paid quite a lot of attention on be sure that we were harvesting berries at the right stage of development. That's a very important thing to begin with, because if you look, if the berry is slightly overripe or if it's underripe, then you have a difference there. And then, yeah, we collected all the berries at the right stage that we found, and we put them all together. So like the sample size was kind of a randomized extraction of berries from this bigger set. How variable that would be, we didn't look at replicates, so I'm not sure I can answer that. We had difference between the 2 years, as I show for the 2 parents, so we know that this trait is quite strongly affected by the environment. I don't know, looking into berries from the bush at the same time, I'm not sure about that. I don't know if anyone has a better answer to this question.
I guess yeah, you answered my question. I guess my concern was in other experimental populations, how do you make the variation within a plant low enough so that you can see variation across the population? And so what you just mentioned there is that the harvesting is really important. And yeah, and that's like the best way to really be able to tease apart the intra from the inter population variation, I guess. So I'll be sure to do that with my stuff. Thank you.
Are there any other questions? Well, it doesn't look like we have any other questions. As I said, we are recording this webinar and we're going to post it. I'll put it just once more in the chat in case you want to review it later on. So here's the link to where it's going to be posted. I just want to thank Sara for a wonderful presentation. Thank you. I know it's very early in the morning there in New Zealand, waking up early and presenting all the data and the wonderful work that you've done there. Thank you everybody for attending. You might be receiving after this survey, if you can provide us some feedback on the webinars, that would be fantastic. We really appreciate getting feedback from everybody and trying to improve how we do things. And with that, thank you everybody, and I hope you have a good day and a good rest of your day. Bye-bye.
Thank you.
Thank you.
Thank you so much.